Hey there, listeners. Welcome to another paper preview podcast. Credit for this work goes to the original authors. Please find a link to their paper on PubMed Central in the description below or using the QR code on screen. This paper preview is meant to be a general overview and introduction to the paper. Please read the actual text and figures for more details. The following discussion, script, and voices were created using AI and are not intended to mimic the likeness of the original authors.
Welcome back to The Deep Dive, the show that extracts the most important nuggets of knowledge from cutting-edge research and brings them directly to you. Today, we're embarking on really a fascinating journey into a discovery that challenges one of the most fundamental principles in cell biology. I'm talking about the long-held belief that cells are entirely self-sufficient, especially when it comes to managing their internal components. For decades, our textbooks have presented the cell as, a self-contained metabolic factory, meticulously managing its own resources and waste. Think of it. Every broken piece, every worn-out machine part handled internally, dismantled and recycled right there on site. Mitophagy, the process of a cell eating its own damaged mitochondria, is a prime example of this. Well, this cell autonomous dogma is a bedrock concept often taught as a completely cell autonomous process where a cell manages its own internal housekeeping, no outside help needed. But what if this isn't always the full story? What if some of these vital cellular waste products, especially in complex tissues like the brain, what if they aren't recycled in-house, but rather exported, processed by a neighboring cell? Okay, let's unpack this. What if a cell doesn't always degrade its own mitochondria? What if there's a more collaborative and, frankly, really surprising dance happening between different cell types, especially somewhere as intricate as the brain? Today, we're dissecting a truly groundbreaking paper, Transcellular Degradation of Axonal Mitochondria, published in the prestigious journal PNAS, which dares to ask that very question. And we're incredibly fortunate to have one of the lead authors with us, the researcher himself, to guide us through this remarkable discovery. This paper's significance, as stated right in the source, is profound. It calls into question the assumption that a cell necessarily degrades its own organelles. Okay. Now, as a journal editor, when a paper like this crosses my desk challenging such an established concept, the first thing I look for is, ironclad evidence. And this paper really delivers. For you, our listeners, especially those of you who are graduate students in biomedical research, our mission today is to explore this paradigm shift. We'll not only uncover the meticulous experimental evidence that supports it, but also delve into the innovative techniques employed. We want to understand the thought process behind challenging such a foundational idea and critically consider what this all means for our understanding of brain health and disease. It's really a masterclass in how to challenge assumptions, design rigorous experiments, and open entirely new avenues for scientific inquiry. So let's start at the beginning. For a long time, we've universally accepted mitophagy as a form of autophagy, meaning self-eating, a cell degrading its own damaged mitochondria. Could you elaborate on what was generally accepted before your work came along and why that concept was so deeply ingrained in our understanding?
Yeah, absolutely. That's an excellent and crucial place to start because it really sets the stage for what we found. The prevailing view, indeed, was that cells are largely self-contained, autonomous units when it comes to organelle quality control. You can picture it like a sophisticated microfactory, meticulously managing every aspect of its internal operations, making new parts, fixing old ones, and recycling the worn-out bits. This idea of self-sufficiency has been a core tenet of cellular biology for decades.
Right. It seems efficient, doesn't it? Keep everything in-house.
Exactly. And mitophagy, as you pointed out, fits perfectly into that model. It's understood as a specialized form of autophagy. Damaged mitochondria get selectively engulfed by these double-membrane vesicles, autophagosomes, which then fuse with lysosomes.
The cell's recycling centers?
Precisely. For degradation. And the term autophagy itself, from the Greek for self-eating, inherently implies the cell is degrading its own stuff. So this concept of cell autonomous degradation was just fundamental, especially in the brain where keeping neurons healthy is so critical. We assumed a neuron managed its mitochondria's entire life cycle, from birth, transport down the axon, to disposal, all within its own boundaries.
Made sense. Logically, you'd think that's how it works.
It did. It seemed efficient, precise, localized quality control. It's what the textbooks taught, what we all learned. But as our research shows, sometimes biology has more intricate, maybe more collaborative solutions up its sleeve than we first imagined.
And what a revelation it is. So, given this deeply entrenched view, what exactly did your research uncover? What was the finding that fundamentally challenges this assumption about self-degradation, particularly in the CNS?
Well, what we found is something fundamentally different and honestly truly surprising, especially in the central nervous system. Imagine that factory again. But instead of dismantling its old machines on site, it efficiently packages them up and sends them next door to a specialized recycling plant.
Okay, interesting analogy.
That's essentially what our research reveals. In normal wild-type mice, we discovered that in a very specific yet critical location, the optic nerve head, or ONH, the axons from retinal ganglion cells actually shed their mitochondria. They literally package them up and release them.
Shed them, not just degrade them inside.
Exactly. Shed them. And here's the kicker. These shed mitochondria are then internalized and degraded by adjacent glial cells, specifically astrocytes. We actually coined the term transcellular degradation of mitochondria, or transmitophagy, to describe this novel, non-cell autonomous process.
Transmitophagy. Okay, so trans meaning across cells.
Precisely. It was a complete rewiring of our understanding of how these specific components were handled. And the location, the ONH, is fascinating. It's a highly vulnerable spot, structurally and metabolically.
That's where all the axons from the eye bundle together, right?
So exactly. Over 1 million retinal ganglion cell axons converge there, exit the eye, and crucially, they transition from being unmyelinated to myelinated. So it's under immense metabolic stress, mechanical stress. It's a key site for understanding neuronal health, and also diseases like glaucoma.
Ah, okay.
So the profound implication here is that this discovery directly questions that long-standing assumption that a cell necessarily degrades its own organelles. It points towards a collaborative effort for keeping neurons healthy, not just an isolated cell-autonomous job.
It really changes how we think about glial cells too, doesn't it?
Absolutely. It opens up entirely new ways of thinking about quality control, how cells talk to each other, glial neuronal interactions. It means astrocytes aren't just passive support struts. They are active partners in keeping neurons functional.
This is where it gets really interesting. You're talking about shedding and engulfment. I mean, that sounds incredibly dynamic, visually striking. Given that prior dogma, how did you first see this happening? What were the initial, maybe puzzling observations that made you even suspect something other than standard mitophagy? It must have taken some incredibly careful observation to spot something so unexpected. What was that aha moment like?
You're right. Seeing is believing, especially for something this novel. And the visual evidence, particularly at the ultrastructural level, was absolutely paramount. Our journey actually began with very detailed electron microscopy. We weren't specifically looking for transmitophagy initially. We were more focused on understanding general mitochondrial dynamics in the ONH because it's known to be vulnerable, like in glaucoma. But sometimes, when you look closely enough...
Biology surprises you.
Exactly. So to really see this, we had to push the limits of microscopy. We used a powerful combination of techniques. We started with serial block phase scanning electron microscopy, SBEM.
Right, that gives you the big 3D picture.
Precisely. It's like automatically slicing the tissue into incredibly thin layers, nanometer scale, imaging each one, and then computationally rebuilding it into this vast 3D volume. That gave us amazing spatial context, let us trace processes over long distances. Essential for understanding the architecture.
And then for the fine details.
Then, for the really exquisite detail of what was inside these structures, we used high-resolution transmission electron microscopy, TEM, and even EM tomography.
Tomography, like a CAT scan, but for tiny cell parts.
Kinda, yeah.
Okay.
It takes multiple TEM images from different angles to build an even more precise 3D model of individual components. It was this multi-pronged ultrastructural approach that first revealed these large, distinct protrusions coming off the axons. What we initially saw were quite striking, these large membrane-bound buds or protrusions, originating from axons that otherwise looked perfectly healthy, right there in the ONH.
So not from damaged or dying axons.
Not necessarily, no. They looked otherwise healthy. And these weren't subtle little bumps. They were prominent structures, almost like little balloons budding off. When we looked inside these protrusions and the subsequent avulsions...
Avulsions meaning the bits that pinched off.
Exactly. Membrane-enclosed packages that have pinched off, like self-contained vesicles. We consistently found organelles with the unmistakable features of mitochondria. You could see the cristae, everything. What was really striking was seeing both intact mitochondria and fragments of mitochondria within the same avulsion. That suggested, maybe a process of ongoing degradation or packaging of damaged goods.
And where are they located specifically? That seems key.
That was the start of the aha moment. They weren't just randomly scattered. Crucially, they were found specifically, consistently, right at sites of direct physical contact between the axon membrane and the processes of astrocytes.
Ah, the astrocyte connection.
Yes. And as we followed these in our 3D reconstructions, we saw these membrane-enclosed avulsions still packed with mitochondria, becoming fully surrounded by astrocyte cytoplasm, like the astrocyte was engulfing them.
Wow. Okay.
And EM tomography, with its superfine detail, showed they weren't just empty bags of mitochondria either. We saw microtubules nearby, meaning a bit of the exoplasm was being shed along with them.
So it's a chunk of the axon's contents focused on mitochondria.
Essentially, yes. And to give you a sense of scale, we quantified this meticulously. In 3- and 9-month-old standard lab mice, C57BL6J, we found densities of around 1.9 and 4.8 of these clusters per 10,000 cubic micrometers, respectively. So it increases with age.
That's quite a few.
It is. And each protrusion was pretty substantial, average volume around 11 cubic micrometers, containing on average about 29 mitochondria plus or minus 18. So this wasn't some rare one-off event. It was consistent, observable, and quantifiable. This detailed EM work was our first really powerful line of evidence. It provided these static snapshots that painted a compelling picture of transcellular transfer.
And for the grad students listening, a real testament to the power of just looking carefully with the right tools.
Absolutely. Cutting-edge imaging and just perseverance and careful observation. Sometimes the answers are right there if you have the resolution and patience to see them.
That's truly remarkable. Seeing those structures at such detail builds a compelling case for the physical transfer. But like you said, proving they're being actively degraded, and crucially, by which cell, that's another story. EM gives you static pictures, but degradation is dynamic. You needed a live reporter, right? How did you design something to track mitochondrial degradation in real time and really nail down where it was happening, specifically in the receiving cell, the astrocyte?
You're absolutely right. Moving from those static EM images to a dynamic, quantifiable reporter was the next critical step. We had to confirm these mitochondria weren't just being shed, but actively degraded by the astrocytes. Our reasoning was, well, if mitochondria are being internalized and degraded by another cell, they have to end up in that cell's lysosomes.
Which are acidic.
Exactly. Highly acidic compartments for breaking down waste. And that acidity became the key to our reporter design. We developed what turned out to be a pretty ingenious genetic reporter system using a tandem fluorophore.
Okay, explain tandem fluorophore.
So the core idea is using two fluorescent proteins with different sensitivities to pH. Our construct, which we call mito-EGF-PMCID, fuses an acid-sensitive EGFP that's enhanced green fluorescent protein with an acid-resistant protein called MCID. This pair of fluorophores was then specifically targeted only to mitochondria.
How did you target it?
We fused it to the first 29 amino acids of a mitochondrial protein, human cytochrome C oxidase subunit 8. That sequence acts like a zip code, ensuring the reporter goes exclusively to mitochondria within the cell. Then, delivery. We used an AAV2 vector, a harmless virus commonly used in gene therapy, delivered by injection right into the eye intravitreally.
And why that delivery method?
Crucially, this method ensures expression only in the retinal ganglion cells and their axons, the neurons we were interested in. We rigorously checked and confirmed no expression of our reporter in the ONH astrocytes themselves. That specificity was absolutely vital.
So any signal you saw in astrocytes had to come from the neurons.
Precisely. That was the linchpin. So here's how it elegantly reports degradation. When a mitochondrion with mito-EGF-PMCID is healthy, chilling in the neutral pH of the cytoplasm, both EGFP and MCID glow brightly. You see a combined yellow-orange signal.
Okay, both on.
Right. But once that mitochondrion gets taken up by, say, an astrocyte and enters an acidic lysosome for degradation, poof, the EGFP fluorescence is quenched by the low pH. It stops glowing green. But the MCID, being acid-resistant, keeps shining brightly red.
Ah, so you're left with just red spots.
Exactly. Distinct MCID-only puncta. These bright, isolated red spots become our clear, quantifiable signal of acidified, actively degrading mitochondria. And we painstakingly confirmed this worked by showing these MCID-only puncta reliably colocalized with LAMP1, a standard marker for lysosomes, in the retina.
Okay, so the reporter works. What did you see in the ONH?
Now, for the key finding, this is where the dynamic evidence really nailed it. When we looked at the ONH in mice, where only the retinal ganglion cell mitochondria were labeled, with our AAV2.mito-EGF-PMCID, we found an abundance of these MCID-only puncta, the degradation signal.
And where were they? Inside axons or?
That's the critical part, the crucial spatial localization. These degradation signals were not primarily found within the axon bundles themselves. If they were, that would suggest standard cell-autonomous mitophagy.
Right.
Instead, they were robustly localized within the intervening glial columns, the spaces between the axon bundles, which are filled with astrocyte processes. And importantly, they clearly colocalized with MAC2.
What's MAC2?
MEC2 is a known marker of phagocytic activity, cells that eat things. And it's particularly abundant in the astrocytes, right there in the ONH, in what's called a myelination transition zone, or MTZ.
So the degradation signal is inside phagocytic astrocytes.
Precisely. So this leads to a fascinating thought for everyone listening, especially the students. If the reporter is only made in neurons, but we clearly see the degradation signal, these distinct red M-cherry-only dots localized within astrocytes, what does it fundamentally tell us?
It has to be crossing between cells, non-cell autonomous.
Yeah, exactly. It strongly, unequivocally suggests a non-cell autonomous degradation pathway. Material from the neuron is being broken down by the astrocyte. It's direct evidence of transmitophagy happening live.
That's an incredibly clever system, really elegant. And seeing that localization in the astrocytes is powerful evidence for transmitophagy. But, for a discovery that challenges such a fundamental assumption, relying on multiple independent lines of evidence is absolutely crucial. You need that ironclad case. What other ways did your team confirm that these degrading mitochondria, now seen inside astrocytes, truly originated from the retinal ganglion cells? How did you remove any lingering doubt about the transcellular nature?
Precisely. You're hitting on a key point about scientific rigor. For something this paradigm-shifting, we absolutely needed convergent evidence from entirely different methods. Our third crucial piece of the puzzle came from a sensitive assay designed to detect DNA degradation, specifically mitochondrial DNA, or mtDNA.
Okay, looking for the mitochondrial genes themselves being broken down.
Exactly. This involved combining two techniques. The 2N assay, which detects fragmented DNA and is a hallmark of degradation or apoptosis mine, and a technique called mitofesh, which uses fluorescent probes designed to stick specifically to mitochondrial DNA sequences.
So Tunel says DNA is fragmented, mitofish says, and it's mitochondrial DNA.
Perfect summary. The principle is simple. As mitochondria get chewed up in lysosomes, their contents, including their mtDNA, also get fragmented. By combining Tunel and mitofish, we could literally visualize these fragments of mtDNA within cells. And what we observed were distinct clusters of signals that were positive for both Tunel and mitofish. And crucially, these double positive signals were found unequivocally within the cell bodies and processes of ONH astrocytes.
How did you know they were astrocytes?
We could confidently identify them because we used mice where astrocytes were genetically labeled, expressing a fluorescent protein under the control of an astrocyte-specific promoter, GLT1.EGFP. So the green cells were astrocytes. And within these green astrocytes, we saw the red magenta Tunel mitofish signals. This provided direct biochemical evidence that astrocytes weren't just eating empty bags, they were processing actual neuronal mitochondrial remnants right down to the DNA.
That's pretty specific.
It is, but we needed to be absolutely sure. To rule out any potential artifacts, we performed a critical control experiment. We treated tissue sections with DNANC.
The enzyme that chews up DNA.
Right, a potent DNA-degrading enzyme. When we pre-treated with DNANC, any non-nuclear Tunel and mitofish signals were completely wiped out. That confirmed the signals we were seeing were genuinely due to fragmented mtDNA, not some weird non-specific staining. Gave us immense confidence.
Okay, so mtDNA fragments are in astrocytes, but the final link proving they came from the axons.
Yes, the most critical piece of evidence connecting everything came when we combined the Tunel labeling with our AAV2.mitoEGF PMCID reporter system. So now we have mice, where the neuronal mitochondria are labeled red or yellow-orange, and we're also looking for fragmented mtDNA with Tunel. What we found was that the Tunel signal indicating the fragmented mtDNA largely colocalized with an mCherry-only puncta within the ONH astrocytes.
Wow, okay, let me process that. The red-only dots from your reporter, meaning acidified neuronal mitochondria in the astrocyte, also light up with the TNL signal, meaning their DNA is fragmented right there.
Exactly. It's almost undeniable confirmation. The very mitochondria shed by neurons, reported as degrading by our tandem fluorophore inside astrocytes, were also the ones showing DNA fragmentation inside those same astrocytes. So if you connect the dots, these three independent lines of evidence,
the EM showing physical transfer, the live reporter showing degradation in astrocytes, and the DNA assays confirming axonal and tDNA fragments in astrocytes.
Precisely. They provide robust, convergent, irrefutable support for transmitophagy. Each method tackled the question differently, and all pointed to the same astonishing conclusion. Neurons shed mitochondria, astrocytes clean them up, challenging decades of dogma. It really shows the power of hitting a question from multiple angles.
The evidence is certainly compelling, absolutely. That multi-pronged approach leaves very little room for doubt about transmitophagy actually happening. But, understanding a mechanism is one thing. Appreciating its physiological impact, that requires quantification. This process sounds incredibly significant, but to really grasp its importance, we need to understand the scale. Is transmitophagy just a minor cleanup crew for a few damaged outliers? Or is it major pathway? Is it contributing substantially to the overall turnover of mitochondria in these neurons? How did you quantify the scale? What did the numbers tell you?
That's a crucial question, absolutely. Discovering a mechanism is foundational, but understanding its relevance means getting quantitative. We had to figure out if this was peripheral or central. To address this, we developed and rigorously used what we call the mitophagy index, or MI.
Okay, the MI. How does that work?
It's essentially a ratio that gives a quantitative measure of mitochondrial degradation. We defined it as the M-cherry-only I signal, the red-only degraded signal, divided by the total M-cherry signal, which represents all the labeled mitochondria, healthy or not. You can calculate it based on volume from 3D imaging, we call that MiVol, or based on area from 2D images, Meyer area. Depends on the analysis method.
So a higher MI means more degradation is happening relative to the total amount of mitochondria present.
Exactly. A higher MI indicates a greater proportion of acidified, actively degrading mitochondria. First things first, we had to validate that our MI was actually a sensitive and reliable measure. We needed a positive control. We used rotenone. It's a well-known drug that messes up mitochondrial function, specifically complex I.
So it should trigger mitophagy.
Right. When we injected low doses of rotenone into the eye, much lower than doses that kill cells, we saw a dramatic dose-dependent increase in the ONH. Specifically, a huge 16-fold increase in my vol and a 3.4-fold increase in my area. This clearly showed our reporter system and the MI calculation were highly sensitive to increased mitochondrial degradation. We had a reliable tool.
Good validation. So what happened when you compared different locations?
Then came the critical comparison, and this is where the data really started to challenge things. We wanted to know, how does transmitophagy in the ONH compare to degradation happening in the retinal ganglion cell soma, the cell body, back in the retina?
The GCL, right, the traditional place we thought most degradation happened.
Exactly, the GCL. Using sophisticated 3D image analysis software, MRS, we found that the volume-based index in the ONH was approximately 2.7-fold higher than in the GCL.
Higher in the ONH, wow.
Yeah, that was already striking. It suggested the ONH is a really significant site. But we wanted to be even more rigorous, minimize any potential subjective bias from manual image analysis. So we also used an even more advanced method called 2D segmentation matrix statistics, or 2D SMS, for 2D images.
Okay, what does that do differently?
It's computationally intensive but very objective. It analyzes images across multiple segmentation thresholds for both color channels simultaneously. Really robust quantification. With 2D SMS, the difference was even more dramatic. The my area in the ONH was approximately 4.6-fold greater than in the GCL.
4.6 times higher degradation rate in the ONH compared to the cell body?
Per unit of mitochondria, essentially, yes.
Yeah.
And to be absolutely sure, we ran controls with scrambled data, confirming the statistical validity. We were confident in the numbers. Now here's the really profound interpretation, where the full impact becomes clear. Despite the fact that the GCL, the cell body region, contains roughly 10 times more mitochondrial volume overall than the relatively small ONH region.
Right, the cell body is much bigger.
Much bigger. But our calculations using the MI suggest that the total amounts of retinal ganglion cell mitochondria being degraded at both locations, the ONH via transmitophagy and the GCL, presumably via cell-autonomous mitophagy, are actually comparable.
Wait, say that again? The total amount is comparable?
Yes. The sheer volume of mitochondria being cleared in that small ONH region is roughly on par with what's happening in the much larger cell body region.
That's astonishing.
It is. This data conclusively establishes the ONH not as some minor cleanup site, but as a major site for retinal ganglion cell mitochondrial degradation, a critical hub. It fundamentally shifts our perspective. It suggests that for these neurons, a huge chunk of mitochondrial quality control is effectively outsourced to their glial neighbors at this strategically vital spot.
So what stands out to you about those numbers? How does that force us to rethink neuronal metabolism?
Well, it profoundly challenges that ingrained idea that the soma handles everything. It suggests neurons have evolved this incredibly efficient, localized disposal system at a potential bottleneck. Maybe it relieves the burden on the soma, maybe it's faster, maybe it prevents damaged goods from having to travel all the way back down the axon. It forces us to rethink the metabolic strategies and vulnerabilities of neurons in a whole new way.
That quantitative data is incredibly powerful, absolutely reshaping our understanding of where this crucial housekeeping happens. It really paints transmitophagy as a major player. Now, this was all beautifully shown in the optic nerve head, which, as you've explained, is a very specialized, high-demand area. This naturally leads to the big question for neuroscience generally. Is this unique to the visual system, a special adaptation for this bottleneck? Or could transmitophagy be a more general process, happening throughout the central nervous system, implying a widespread reliance on astrocytes for neuronal mitochondrial health?
That's the logical next step, isn't it? An extremely important question for the broader implications. The ONH is special, yes, with unique stresses. But our initial observations and just the sheer elegance and efficiency of transmitophagy made us hypothesize this might not be confined just to the ONH. It seemed too crucial a mechanism to be that localized. So we immediately started looking elsewhere, using SBEM again, to search for similar structures in other, more general parts of the CNS.
So looking beyond the eye-brain connection, where did you look?
We look in the cerebral cortex, specifically in the superficial layers, layer 1, of young, healthy, wild-type mice. And what we found was quite compelling, supporting that broader relevance idea. Our SBEM analysis identified numerous protrusions there too. When we examined these structures carefully, they looked remarkably similar to the mitochondrial-filled protrusions and avulsions we'd characterized in the ONH.
Morphologically similar. Same kind of structure.
Very similar, yes. In overall shape, membrane-bound, filled with debris, including recognizable mitochondrial fragments, often right at points of contact between axons and astrocyte processes. This striking similarity strongly suggests that the underlying biological process, this shedding by axons, engulfment by astrocytes, is indeed shared across different brain regions.
Okay, that's significant, but was it happening at the same scale?
That was the main difference. While they look similar, these cortical structures were found at a significantly lower density, roughly 10-fold lower compared to what we saw in the ONH.
10 times less frequent in the cortex than the ONH hotspot.
Exactly. So our interpretation is that transmitophagy of axonal mitochondria by astrocytes likely is a widespread phenomenon in the CNS. It's not just the ONH. However, it might be less prominent, maybe less constantly active than the intensely metabolically demanding ONH. The ONH might be a hotspot, but the basic machinery seems broadly distributed.
That makes sense. It fits with other known astrocyte functions too, doesn't it?
Beautifully. It connects really well with what we already know about astrocytes being active phagocytes elsewhere. For instance, there's seminal work showing astrocytes phagocytose entire synapses, helping sculpt neural circuits, particularly during development in the visual pathway.
Right, synapse pruning.
Exactly. And since synapses typically contain mitochondria, often strategically placed right there to fuel transmission, it raises a fascinating possibility. It's quite plausible that the molecular machinery astrocytes use for eating synapses might also be used, maybe with some tweaks, for eating these axonal mitochondrial avulsions.
So it could be part of a more general astrocyte cleanup crew toolkit.
Precisely. It points to a more generalized, active role for astrocytes throughout the brain, extending beyond just synapse pruning to include disposal of these larger neuronal components. It suggests a fundamental, maybe evolutionarily conserved way of managing resources and waste between cells in the nervous system, highlighting the brain as this truly collaborative organ.
This research has undeniably opened up so many new avenues for exploration. It's incredibly exciting, especially for graduate students thinking about future projects. It really feels like the tip of the iceberg, revealing there's a whole new layer of intercellular communication and dependency. But, with every groundbreaking discovery, more questions pop up than answers, right? Looking forward, what are the big unanswered questions that you and your colleagues are grappling with now? What's next for this burgeoning field of transmitophagy? What are the really exciting frontiers?
You're absolutely right. It feels like we've just cracked open a door, and beyond it lies this vast, unexplored landscape. That's the exciting part. One of the big unanswered questions revolves around mitochondrial trafficking within the axon itself. We're still trying to figure out, are the mitochondria removed at the ONH just the ones that were already there, maybe reaching the end of their lives? Or are mitochondria from further away, maybe back in the retina, actively transported to the ONH specifically for disposal?
Like a designated waste disposal site.
Exactly. Like a sophisticated commuter train for cellular waste, routing dysfunctional mitochondria to this central hub. Investigating that is tricky, needs complex live imaging of mitochondrial movement along axons. Another huge question, and one we find incredibly fascinating, is why the ONH? Why is it such a major hotspot for this?
Right. What makes it so special?
We have a few working hypotheses. One, maybe transmitopathy gets strongly activated by local axonal damage or metabolic stress, even if it's mild, not enough to kill the axon. A proactive quality control. Two, maybe it's just logistics and energy. Perhaps it's too dangerous, too costly, or too slow for the neuron to drag damaged mitochondria all the way back to the distant soma, especially through that narrow glial lamina bottleneck in the ONH.
So the ONH is just a convenient local drop-off point.
Could be. Or three, maybe the astrocytes there are special. The ONH has astrocytes with extremely high phagocytic activity, marked by things like MAC2. Maybe they're specialized dedicated waste management crews.
Teasing those possibilities apart sounds challenging.
Very. It would need precise experiments, manipulating stress or astrocyte activity right there in the ONH, in vivo. Then there's the clinical relevance, which is a massive focus now. We're urgently looking at diseases like glaucoma.
Makes sense, given the ONH connection.
Absolutely. Glaucoma involves axon loss in the optic nerve, and early signs often include these focal swellings and dystrophic bits on axons packed with mitochondria right in the ONH. So, is normal transmitophagy impaired in glaucoma before the axons actually die? Could boosting transmitophagy be neuroprotective? That's a huge therapeutic question.
And potentially relevant for other neurodegenerative diseases, too.
Definitely. Given how critical mitochondria are for axons and their known dysfunction in Alzheimer's, Parkinson's, ALS, we absolutely need to explore if transmitophagy defects contribute there too. That means looking at animal models, maybe even human postmortem tissue.
What about the how, the molecular nuts and bolts?
Yes, on a molecular level, we're very keen to identify the specific machinery astrocytes use. Are they using the same receptors and pathways as for synapse eating, or are there novel mechanisms? Finding those molecules could give us drug targets to modulate transmitophagy. Are there eat-me signals on the shed mitochondria? Find-me signals from the axons? Big open questions.
And you mentioned parallels elsewhere in biology.
Right. It's fascinating to consider the evolutionary angle. This kind of intercellular cleanup isn't unique. Transmitophagy happens in fertilization. The egg cell degrades the sperm's mitochondria, ensures only maternal mtDNA gets passed on.
Wow, okay.
And in the eye, retinal pigment epithelial cells constantly chew up the shed outer segments of photoreceptor cells. Another classic example of one cell type disposing of another's parts. So it suggests this might be a fundamental, conserved biological strategy, managing cellular components across cells. And finally, the immunological angle is critical. Mitochondrial DNA is highly immunogenic. If it leaks out because transmitophagy fails,
It could trigger inflammation.
it's exactly chronic inflammation contributing to disease. Studies have linked mtDNA release to things like cardiomyopathy. So efficient transmitophagy isn't just housekeeping, it's actively preventing harmful inflammation. The questions are just boundless, from basic biology to clinical potential. It's a really exciting time to be digging into these intercellular dynamics.
That's an incredible look into the future, truly. A testament to how one breakthrough can just ripple outwards, creating so many new research frontiers. So, wrapping up, what does this all mean for our broader understanding? We've really moved, haven't we, from that rigid, self-eating model of the cell as an isolated island to seeing this sophisticated, collaborative, interdependent process, especially in the brain. It's a fundamental reevaluation of how neurons maintain mitochondrial health, and it highlights the critical, active role of astrocytes. They're not just support cells anymore, they're indispensable partners in neuronal maintenance, maybe even neuroprotection. Truly the unsung heroes of the brain's waste management system, it seems. For you, our graduate student listeners, this deep dive really underscores the importance of challenging those long-held assumptions. Even bedrock concepts can be revisited. It showcases the power of meticulous experimental design, from the beauty of EM to clever reporters to rigorous quantification. And it highlights the huge translational potential. Basic science like this paving the way for new ideas about treating devastating diseases. It really encourages you to think beyond single cells, to see that intricate web of intercellular dependencies. Don't be afraid to ask what if, even about the dogma.
And maybe the final thought to leave everyone with is this. If neurons rely on their glial neighbors for something as critical as mitochondrial disposal, what other hidden or outsourced intercellular dynamics are at play in brain health and disease that we haven't even begun to uncover yet? It's a powerful call to look beyond the obvious, embrace complexity, and stay endlessly curious about the subtle, profound collaborations that keep us all functioning.